Hi, everyone. It's my honor to introduce Dr. Kith Pradhan. Dr. Kith Pradhan is a assistant professor in the Department of Epidemiology with expertise in statistics, bioinformatics, and computing on large-scale omics data. He graduated from Stony Brook with a master in computer science and a PhD in applied math and statistics. He then went on to complete a postdoc here at Einstein. After working at Cold Spring Harbor Lab as a scientific informatic developer for a few years, he joined Einstein again as a faculty, working with the lab of Dr. Amir Vermont as a bioinformatician. He now also teaches courses such as QSBR3 for people who are highly interested in statistics, omics, as well as RNA-seq. I will highly recommend you to take the course. So without further ado, we will begin the session with Dr. Kith Pradhan.
So there should be a link at this omics website under the number 3, specific vignette instructions. And if you scroll all the way down, there's a link to a Dropbox folder. And I sort of hope that you will read the email and you download this beforehand, because it is a somewhat large file. And especially, it's the preparation or the installation of the libraries. That's going to really take the most amount of time if you're going to be following along this tutorial interactively. So I just hope that you've done that beforehand. And if not, now's the time to start installing libraries, because we're going to go over like a little presentation, like an overview of like what this workshop is going to be about. And during that time, that's when you want to like have the computer like installing. Like, so it might take 20 minutes, it might take 10 minutes. If you already like did this sort of analysis before, and you might already have these libraries installed, but you do need to have the libraries installed to be able to run this code. And it's just something. Sometimes this error, like a lot of people have problems installing packages, and really now's the time. Like if you have questions, maybe we can help you out. But to do any sort of work or any sort of analysis in R, like using these packages, like you really need to have them installed. So let's see. So we'll go right to the zip file. If you unzip it, you'll have sort of a directory, like a structure that sort of looks like this. And in it, there are the 3 main files are these 3 HTML files. There's tutorial 0, tutorial 1, and tutorial 2. Just if you can, everyone open up that first tutorial, the prerequisites. This is just like an overview of how this workshop is going to be run, where we have 2 main workshops. One is for RNA-seq, one is for ATACseq. And in both of those tutorials, there's going to be 2 sections. The first section is going over like how you actually process these RNA, like the FASTQ files, how you process the data to get it into a form where you can actually run the statistics, run the analysis. And that is generally, it takes a long time. And generally, you're going to be using the HPC, like high performance computing resources, to do this thing, because most times like your laptop isn't really strong enough to run this sort of alignment and all these other procedures. So basically, the first part is just going over the commands that you would run to actually process the data. And then the second part is where it's like an interactive session where we're going to be using R and going over like the already processed data and how to like analyze it, how to do this differential expression analysis, and how to do all the other analysis for this omics data sets. So the first part is just we're not actually going to be running this processing, but I am going to be going over the code. Just like it's mostly like bash shells, like bash scripting, going over the HPC commands to actually get the data submitted in your Slurm manager. The second part is we're going to be downloading a small data set and working with that interactively, and hopefully you'll have your R Studio or your R like ready on your own laptops, and you can like work along with me interactively. And I really hope that you do work on this code interactively, because it's generally like you just learn so much faster when you're actually running the code yourself, or even if you're just copy pasting the code, when you like can see what the code is doing and you see like the output of that code like right away, like I find it's so much easier to learn than to just sit there and just try to follow along on a presentation. I would like each of you, if you have your R installed, just try to run these packages. Just if you open up your R, If you have all these packages installed, that's great. You should be able to run the actual code no problem. If not, if you're missing some packages, try to like just install them. This code below is just like some code. You can pretty much copy paste this code into your RStudio and it'll install the missing package. Like if you're missing a library, it's gonna like find which ones are missing and just install those ones. You might run into issues though. So I suggest you start that now if you haven't done it before. And in about 10 minutes, we'll actually get to like the actual tutorial. And there's also one more thing. If you go to section 3, there is one thing we're going to be doing at the very end, where we're just going to be visualizing these coverage files from an ATACseq analysis or like a data set. And these files themselves, they're really big. I think it's about 600 megabytes. So if you want to try to do this part interactively with me, I suggest you start downloading it now. So by the time we get to that part at the end of the lecture, it's going to be like an hour or two. Hopefully everyone would have this data already downloaded. So this is the first section. Just try to get all these libraries installed, because I can pretty much guarantee if you can't get this part to run, you're not going to get the other code, the actual code, the tutorial code. Yeah. So if you open up your R, like, do you use RStudio or do you use like how do you R? Like, what's your preferred method of? Right. So usually you have some sort of IDE. Probably the most useful or the most common one is RStudio. And what you have to, because you have this R is like a programming environment, a programming language, and then you have this IDE, RStudio, which is sort of attaches itself to this environment. So you basically you do your programming, you do your analysis, or you pretty much work with your code through RStudio through an IDE, and then the R itself is just what runs in the back. It's like the environment that all this code is running in. So you need to have that. If you go to the RStudio website, if you just go to Google, type RStudio, you should be able to find instructions on how to download that IDE and also how to download R. But generally, once you have R installed on your computer, installing a package is done with a command called install packages. Basically, it's install that packages and then the name of the package. Like, for example, if you want to install DSeq2, actually, DSeq2 is a bioconductor package, so it's a little bit different, but most packages can be installed by command install that package. They have the packages that are sort of part of this other repository, this bioconductor repository. And if you want to install packages from there, it's a little bit different. You actually have to install this function, this bioconductor biomanager function, and that sort of takes care of installing packages for you. So basically, if you can just copy paste this code into your R, it should start the process of installing all the code. So if you copy paste this, it's going to install all the bioconductors. This is the actual code that does the installation of the bioconductor. And the problem with this is that bioconductor packages, they have so many dependencies, especially like DSeq2. That one, it might take like half an hour to install all the packages. And there's always problems like maybe like Java is installed, maybe it needs something else. So it like generally this is the part that like causes the most headache. So prefer I sort of hope that you would sort of get to this part before the lecture, but we still have time. So hopefully maybe half an hour, you should be able to get some of these libraries installed, or hopefully all of them. But basically this is the code that you would copy paste into your R to start the installation process. This is the code for bioconductor, and below, this is the code to install all the missing regular packages, the R packages. But in general, it's install.packages is how you install a typical R package. So any other questions before we begin? So I'm just curious, who here is running RStudio like on their laptops? And who here is running like who here was able to get into the cluster, the HPC cluster? So yeah, who needs help? So maybe so I'm going to be going over like a short presentation that's just pretty much an overview of like what's going on with this workshop. And during that time, hopefully we're going to be coming around to you. If you need help, raise your hand, and we're going to try to sort of go to your conclusion, try to get these packages installed while we're sort of going over this basic presentation. Yeah.
If people do need like technical help or can't figure out what's going on, please maybe just come back here. That might be the easiest way. So we're gonna go. Yeah, people are like totally lost. You can come back here. Thank you.
So I guess at least, or if you want to, just at least try to start downloading these TDF files, these coverage files, if you haven't already, and then just have it downloading in the background. Just so when we finally get to that part, we'll actually have data to work with. So here, I'm just going to open up this this slide. This is a basic presentation or like a basic overview, really just to buy time, I think, to so everyone is able to get all these packages installed and sort of work out their kinks with their laptops. But this is just like a basic outline of what we're going to be dealing with, and just an overview of how omics data, like the specific omics data that we're working with, is. So this first slide is just generally when you have data that's in a very easy to use format. I hope that we all know how to work with this. Like generally you have a spreadsheet, you have like a text file that has a matrix of all your count data. Generally, once it's in this form, it's sort of like straightforward to do the analysis. It's getting to this part that takes the most amount of time, that takes all the processing. And that's what we're going over during this presentation. So generally I think there's 5 main steps to do this sort of analysis. The first is generating the FASTQ files. It's actually generating the sequencing data. The second is pre-processing that FASTQ, the raw data. Then there's alignment, mapping them to like a known reference genome. The fourth one is just feature counting. This is where you have your genomic locations, like your genes, and you want to see how much data, how much RNA molecules, or how much sort of signal is mapping to specific genes. And then finally, once we've done all that, then we actually have our processed, our clean data matrix, then we do the analysis. And that's what the main lecture is going to be about today. So for the first one, just generating the FASTQ file, generating the raw data. The difference between like these next-gen sequencing and just like the previous one, the previous technology, is that we're actually building the sequences like base by base. So we're actually getting an actual, like we're reconstructing these bases, and we know exactly which bases are being sort of generated like from actual data. And generally there's this machine that sort of is able to get these sequences of the bases, ATACseq, and also some sort of measure of quality along with them. And generally they're stored in these FASTQ files, which is the basic text. It's basically a text file that has for every fragment, for every piece of material that you have, it's just like the bases that are sequenced, and then some sort of measure of quality for each of those bases. And that's really your raw data. And if you ever opened up one of these FASTQ files, it's all it is. It's a text file that has basically just that information. It's just there's a line for the name of the fragment. There's a line for the sequence that was actually constructed, and then you have a line that has the quality scores for each of those bases. And once you have these FASTQ files, then there's always a step of just cleaning it up. Like sometimes like you have to trim the adapters, because when the data sequence, you have to sort of add this little piece of this sort of adapter so it can actually stick to the flow of the flow cell, just so you can actually start signaling it. And most of the time you have to break or you have to sort of remove that part before you actually do your processing. There's always like a section of cleaning the data, because a lot of times like the machine, sometimes there's mistakes in it. You want to remove some reads that just have like really low quality. You also want to make sure that if you're doing paired end reads, you want to make sure that the pairs are actually mapping up to each other. So there's always a step for doing this sort of pre-processing. The third step, the alignment, this is the most interesting, and this is usually the one that takes the most amount of time. And the usual way this is done is that you have a big reference library or a reference like genome. And this is just like, you think of it, it's like a huge string, like almost like 2 billion bases of like the T's and G's. And then you have like these little small FASTQ fragments, and you want to see where those fragments are mapping along this huge string. And typically, you never do this yourself, because it's a very complicated thing to sort of be able to map these millions of or even billions of fragments to this like large string. So generally you use software or like programs that have already been written, already been optimized. So generally like use some program like, and I prefer like a star aligner or even like BWA. There's so many, probably like 10 or 20 main aligners that people use. So generally you want to use some sort of like tool that's sort of been developed already. You generally don't want to do this yourself, but there's typically 3 ways to do this. One is just mapping your reads to a huge reference genome. Another one is doing de novo alignment, and that's when you, like, usually this has to be done at least once, like just when you're actually creating the reference. But this de novo alignment is when you have your fragments themselves and you sort of map the fragments to themselves. So like you're building up your own reference genome from these little small pieces. And this one is generally like really, it takes a really long time, a lot of computational power to be able to like build this reference genome from scratch. So it's generally not done for the large references. Well, it had to be done at one point, and that's usually, this is how the actual references that we use, these are like the HG38, the HG90, like this is like how these references were built. There's also another form of, it's called pseudo alignment, where you're, instead of mapping to like a huge reference gene, you're mapping to like specific transcripts. Like it's a smaller pieces of that huge reference, and you're just mapping your reads to the smaller pieces. And this is generally a lot faster than doing either of the other 2 methods. So once you have your data aligned, data mapped, usually so something you always want to do is you want to visualize it. You just want to make sure that you didn't make a mistake, or you mapped to the correct genome. It's just like another source of like just quality control. You always, like, almost like a sanity check sometimes. You always want to load up your data, make sure that your data makes sense, because oftentimes maybe the machine was like, maybe there was a problem with the machine. Maybe you just have garbage reads. You want to be able to visualize this just to make sure that you're actually dealing with something that has signal. And what I like to use is this program called IGV. And you can do, if you just do Google IGV, you'll go to the website. It's a free software. You can even run it in a web browser, but generally you want to install that on your computer, run it in the desktop mode. And whenever you're dealing with like aligned data, it's like, I can't think of a reason why you wouldn't do this, but just view it, visualize it, just to make sure that you're actually like working with non-garbage data. So the 4th step is that once you have your reads all mapped to where they belong in these reference genome, you want to count this, like you want to do some feature counting. So if you're doing like RNA-seq, you want to sort of figure out how much read was, or how many reads or how many fragments are mapping to a specific gene. And this is where you have some sort of database that tells you where these genes are in this like reference genome, like a chromosome position of the gene, like a chromosome start and a stop of like basically a subsection of the big genome, to see like where a gene lies. And then you want to see how many of those reads are mapping to that specific section. And this is also another step that you generally don't want to do yourself. You want to use some pre-existing software, like some code that's been developed, because there's a lot of things to consider when you're sort of mapping these features. For example, like you might have reads or fragments that sort of overlap between 2 different genes. You might have a gene or a fragment that maps to multiple different genes across the genome. So you want to be able to have some software or some sort of routine that sort of accounts for all these things. And usually the software, like the big names in the software for feature counting, like ATACseq and others, they've already considered this. They already like thought about this, and like they have these default settings that generally take care of, like they sort of take the thought out of all the problems that can go wrong with the sort of feature counting. And then finally, once you have your data, once you have your matrix of your sequence counts, that's when you actually get to the analysis. And just an example, this is from a different course, but like as an example for RNA splicing, I found a paper, this is like a review, there's like, I don't know, 10 different software, 10 different applications that are like all written in different languages. And this is just like for RNA splicing. There's like whatever you want to do, there's probably like hundreds of software, hundreds of programs, hundreds of applications out there that are involved in like doing specific analysis for the data that you're working with. For splicing, it's like 10, 11. This is just an example, and what we're doing here today is just going over one specific package, DESeq2, to do a differential expression analysis, and also we're going to use DESeq2 to do an ATACseq analysis. So we're going to be combining this sort of ATACseq peak detection routine to work with ATACseq data, getting to a format like a feature counted format, and then use DESeq2 again to do the differential expression analysis. So this is like an overview of the tutorial, and now we're actually going to get to the tutorial now. So before we get to that part, does anyone have questions, or does anyone have any problems that we might want to address about running RStudio, running R? Any other questions? If not, then we'll get right to the lecture. So in that, oh, actually, maybe before we start, there's this, maybe this is important. So who here has actually opened up a FASTQ file, like one of those raw sequencing files, etc. And when I started out, like I think like all this raw data coming from a machine, it's kind of like overwhelming, and it's like sort of scary to deal with like a FASTQ file. It's like this machine is spitting out all this complicated information. But really, if you open it up, you can just open it up in a text editor. It's really a text file that has information on these fragments that are sequenced. And here is just two ends of two mates of a single sample. This is just the first 3 reads. And basically every FASTQ file just has this format where there's 4 rows that make up information about a fragment. The first row is this big, weird, complicated symbol. This is just an identifier, a unique identifier for the actual fragment. Sometimes they have information on the lane, maybe like the date that the data was generated. But basically, you can think of this just as a name, a unique identifier. The second line is always the actual bases that were sequenced, the A's, C's, T's, and G's, just the string that represents the actual fragment. The third line is always just a plus symbol, just I don't know why they chose plus symbol. The fourth line is just it's the quality score. It's basically a mapping from ASCII symbol to some sort of measurement of how confident you are in the call of the space of each specific base. And basically, so a fast, like an actual fragment, it's always just composed of four rows in this FASTQ file. And generally you have paired end, a lot of times you have paired end sequencing. So you have 2 ends of a fragment that were mapped together, and they sort of go together, and they're paired. And generally they have the same name. If you notice, like this identifier, and I'm just switching between these two. It's the same name. Just if you scroll down all the way to the end, there's just one is like read pair end, or read end 1 and read end 2. So really the only difference is that one has a 2 on the end, saying it's like the second pair, and the other one is the first pair. So this is your raw data. This is like a text file. This is just what comes out of the sequencing machine. And you can just think of it as just little tiny strings, little tiny pieces of fragment. Another thing to look at is just like the actual reference. And this is just what it is. It's like this is a section of chromosome 1. This first part is just N, means like nothing. It's just like the beginning of, generally the ends, the middles, and the ends of a chromosome in the beginnings, they're generally noisy, so we don't really have information on them. But you scroll down a little bit more, this is the actual chromosome of the first. This is actually the basis of the first chromosome. And this is obviously not the full human reference. This is just like a section of the first chromosome. But basically, it's just it's not really. If you just open it up, it's not really overwhelming. It's a string. It's just it's text, and it's like you can think of it as it's just raw, like it's a string data set. And what we do is basically the alignment part of the procedure. Like what we're doing is mapping these tiny little fragments to that large. Like we're mapping a small string to a large string, and that's what the alignment process is. And if you here I can open this up. So this is like an example of what you end up with when you sort of like do this mapping. So here, this is a section of an aligned file where you have these like millions of small fragments, and they're mapped to different sections of the large string. And this is a SAM file. So basically, it's just a series of it's also text data, but it's a series of columns. Each row is a specific read, a specific fragment. This first one, this like complicated string, this is just the identifier. This is like a unique identifier for that specific fragment. We have some like a band flag representing like some information about what went on in that mapping. This is the position where it actually was mapped to. So it was mapped to, this fragment was mapped to chromosome 1 at this position. This is like a cigar string that shows like the general like how that mapping took place. Sometimes you have like insertions, sometimes you have deletions in it. This is just like a general string showing like what went on in the mapping. Then you have a column for like the actual fragment, the sequence that was run. You have column for the qualities of each base in that sequence, and then some other information about like just mapping things that went wrong, interesting features of how that read was mapped to the reference. But again, this is just it shouldn't be too scary. It's really just data, and it's like data that just shows how that fragment was mapped to the huge reference. So another thing to look at is a GTF file, and a GTF file is used for doing the feature counting. So this is where you need to know along like the reference, you need to know where the genes are. And you can get this information from like a GFF file, a GTF file. This is just basically a database that shows where the genes start and stop. So you'll have information on the chromosome, where the starting position along that chromosome is, where the ending position is. If you're looking at a gene, maybe it has information on the exons of the genes, where each individual exon starts. It shows like how the sort of exons like combine and interact with each other. But it's basically information on the actual. It's a database on the features that you're interested in. And there's software that sort of takes in a file like this, a GTF file. It takes in an aligned file and sort of uses them together to get your features, to sort of see where these reads are sort of falling along these specific chromosomal positions. And I think with that, we can get to the lecture now. So in that zip file, there's an HTML file called tutorial1_rna.html. And this is like it's written in R Markdown, and it's basically like personally, like when I learn R, when I work with R, I'm always like just Googling. I'm always like looking at like blogs, I'm looking at like tutorials, vignettes. And for me, like it's the easiest way to learn. It's the easiest way to work with R. So you have like some sort of like reference or some sort of like tutorial that you have code that you hope would work, working code. And then what I do, I generally like follow along the tutorial, and I copy paste like little sections from that tutorial to my own version of R. And then I'm just like working along with the tutorial, like line by line. And I hope that when we get to the part where we're actually doing the R code, I hope that you'll do that too. When we get to the actual like the R part. So I think a lot of times like you'll run into these some snippets of R code that may seem complicated at first, but if you can just copy paste them, you know that it works on your computer. You can sort of like use that as a starting point, and then once you have code like you know that's working, maybe you can like break it down. Like you maybe go to like sort of. We'll get to it when we get to it, but I hope that when we get to that part, you will be running this interactively with me. But in the beginning, we're just going to go over that first section where we're actually going over the processing, like how we get those raw RNA-seq fragments, the FASTQ files. How do we get them into like a form that actually you can work with, you can run and analyze with? So and that's what we're going to start off with that, and then we're going to get to the interactive section. So this part of the sort of vignette is basically just going over these steps. And this is something that generally you run on the HPC, because especially the alignment part, it takes so long, especially if you don't have a very strong laptop or desktop. This is something that most of these steps, you're going to be wanting to use the HPC resources, just because it's just too much when you have data like this. So there are steps that you can run on your own. Like for example, like the quality control. Generally, you have these FASTQ files, and you wanted to see like, is the data, like, is there mistakes in data? Is the data messy? Is it clean? Generally, you use a program like, I'm more familiar with this program with FASTQ, but there are other sort of quality sort of software. There's other quality software that can sort of like give you like reports on how your data is. So in this Markdown file, you'll notice these like blue links, and these are just you can just like click on them. It'll take you to like if I'm sort of describing where, if I'm working with if I'm trying to go over like a software, if you click on this, it'll take you to like a site that we can download the software or get more information about it. So if you notice, like there's some like keywords here and like throughout this tutorial, but just feel free to like just click on them and you get more information. Maybe it'll bring you to the manual. But for this, with this step, generally doing quality control is just, it's very simple. If you have a program called, like, well, I'm more familiar with fescue, but you just do fescue and then the name of the files. It's something you can run on the command line, and it'll generate a report for you that shows like an overview of like the quality scores of these fescue files. But generally, this is it's like an easy step if you know the software to use. And fescue is one of those software that's easy to install. I think it's already installed on the HPC, but this is something that you can run on your own laptop just to get an overview of like the quality of your data. Another step is that for the pre-processing is that you're gonna have to trim the adapters off of your raw sequences. And this is also something that you would use existing software to do. I use something called Trim Galore, and it has these, for me, like the default options or the default parameters usually do a good job with for the sequencing or the data that I work with. You can even use it in paired mode where you just mention that your data is paired data. You give it the different mates of your raw fescue files, and it'll go over it. It'll match. Like sometimes you might have fragments that are missing mates, or maybe like there was they might have a fragment that just had low quality reads all along. So it'll sort of it'll filter out those bad quality reads for you, and the reads that don't have matching mates, and it'll sort of get you to a point where the data is in a somewhat clean state where you can do your other downstream processing. So for alignment, this is where I actually suggest an aligner called Star. It's very fast, but the problem that I had with it is that in the past, why I didn't use it before, we switched over to Star, but it just has such a large memory requirement. I think you're going to need, if you're going to be using Star, you're going to need to be using the HPC resources, because just to do a typical alignment to the human reference, you probably need like 20 gigabytes of RAM to do this sort of alignment. But it's fast though. It's very fast. I think it indexes or does some sort of hashing, but whatever it does, it's very fast, but it just has a really huge memory requirement. So what I was using before that is Top Hat 2. It's like a very old software, an old aligner, but it's something that it doesn't have that huge memory requirement. You can run it on your laptop. This is something you can probably run it with like 4 gigabytes of RAM. And just something to note that whatever, I mean, this is just 2 of them, but there's so many aligners out there. There's like a BWA is another common one. There's just so many, and whatever aligner you use, you're going to want to sort of read that aligner's manual, because there's every aligner has their own sets of parameters, own sets of options, own sets of like little caveats and like little tricks about like how you want to work with it. So always check the manual, always check the documents that came with the aligner. The example that we're using today is going to be using Star and how to run Star specifically on the HPC. And there are some quirks about Star on the HPC that you probably want to know the details of to be able to run it, because this is something that I've struggled with this for a long time, and I finally figured it out. But if you're going to be running your own analysis, own processing, you're probably gonna want to know those specific details. So finally, like feature counting, I'm more familiar with this program called ATACseq. And I think that's probably one of the most popular ones, but there are others. But you can click this link, you go to the HTC website, you can get more information about how to work with this and how to run this. So let's see. So that's just an overview of like some of the programs that we're using. The actual running procedures. Well, this sort of pipeline, it's this procedure. I'm actually using Star for a lot of the pre-processing, because Star has certain like built-in features that maybe they're not well documented, but they have features that like just make the whole thing easier. One thing is that it allows for something called soft clipping. So it's sort of like if you have a fragment that maps to a section, or only maybe you know like a fragment maybe is 100 bases long. Maybe the last 20 bases don't map, but like the first 80 bases. If you allow soft clipping or you use Star with a soft clipping feature, it's able to find a proper match, even though there's so many mistakes. There's 20 mistakes. It's able to like sort of just clip off the end or the beginning of a fragment and sort of like get a valid like alignment without sort of including all those mistakes. And with soft coupling enabled, you can sort of skip a lot of times you can skip the step of doing the adapter trimming. It also has another step called quant mode, which sort of, if you give it a GTF file, it'll do the feature counting for you. So already built into with the alignment, it'll also run this feature counting. So it'll give you a data set, a GTF file that has the chromosome positions of every gene that you're interested in. It'll automatically count how many reads are falling into those regions. So we'll get to actual HPC script, but first, just a little note on running it. So usually you need to, like, unless you have like just a few samples that you want to run in an interactive prompt, generally you're going to be wanting to use the sbatch command to submit these batches onto the HPC. And what I do, I have a script where I sort of generally make a script that does what I want, and then I just call that script with a certain input arguments, with the command line arguments, and then I just submit that to the Slurm workload manager. And generally, when I'm working with a project, like the actual commands that are actually submitted, it might look something like this, where you have like sbatch, the name of the script, and then the parameters of the script. And this script, it has 3 parameters. One is the name of the actual the result that you want to, well, like what your sample is called, and then the 2 paired mates of the FASTQ files. So there are 3 parameters to a script. And generally, if you're dealing with a lot of samples, you'll have like what you're actually submitting to the HPC. It's going to look something like this. But just know, like, this is something, it's not something you want to write by hand. Like generally, like these FASTQ files, when they come from the sequencer, they have all these like complicated symbols. Sometimes they have like, well, this is it's not too complicated, but sometimes they have these weird symbols, and you can't really. It would take like probably like half an hour to sit there and like make sure you're typing every one of these correctly, and there's just too much room for error. So generally, you don't want to do this by hand. You want to write a script that will sort of prepare these batch commands for you. And what I like to do, I like to do like a bash script. So, but I mean, some people are more familiar with, you can do this in Python, you can do it in R, you can do it in Perl, but you're going to want to use some sort of scripting language to do this batch submission, because it's just too much to have to type out by yourself. And it's too easy to make these mistakes. So here, this part of the tutorial, it's just a little script that basically loops through all the files, like FASTQ files in a directory, and sort of just extracts some sort of information about those strings. And it basically just prepares a script to be submitted to the HPC. I guess if this is your first time seeing Bash, this code might seem a little bit tricky or complex, but if you just think of it, it's basically a loop. It's looping through all the first mates of a folder that has all the FASTQ files, and then it's doing something with each of those files. This thing here, it's sort of like the name of the FASTQ file is stored in this variable called FQ1. FQ2, basically it's just a string of pipes commands, bash commands that sort of extracts or sort of finds the name of the second mate. It's just generally, when you're dealing with data, if it comes from like the WASP's sequencing facility here at Einstein, or maybe you're downloading data, maybe like wherever your data comes from, generally you're going to be wanting to look at all this data, all your FASTQ files, and then see if there's some sort of relationship by looking at the strings to see how you match the first mate to the second mate. And usually sometimes it's _1, maybe the first mate is like always an _1, maybe the second mate is always _2, maybe it's like R1, R2, but you have to sort of figure out how these 2 names are related. And if it is an easy like string substitution between the first and the second mate, it's like you can use these like bash commands to sort of just translate them and actually get the name of the second mate. So it saves having to type out whatever. You have 100 files that you have to map. It saves having to type out that long, complicated file name and potentially make errors. But basically, it's just this is just like getting the file name. This is extracting a section of the sample from the file name, and basically what it is, it's just echoing. It's just printing out the command to a text file. Here it's not actually in the loop. It's not actually running the command or submitting the command. It's saving it to a text file. And the reason I do this is just because it's so easy to get these to make mistakes, even when running this code. So generally, what I like to do, I like to submit all my commands to a file that looks like this. Give it a once over, like just look it over, make sure like the script actually did the correct job, and sort of like calling the names, extracting the sample names. And then once I know that it's sort of like makes sense, then I just run the script to the HPC, and like this, like then you just pretty much run this file. You submit all your jobs, and then you wait whatever hour, 2 hours, 3 hours for the data to come in. So all that, there's like some links here. If this is completely new to you, you can like click on these like little hyperlinks, and it'll tell you like what all these symbols are doing, like redirecting, like doing like maybe like piping commands together. Like if this is like your first time seeing code like this, you're probably gonna want to do some research, or like just if you want to understand more, there's some links that sort of explain what all these things are doing. Now the actual script, the code that actually does the actual like processing. This is an example of what a batch submission job would look like on the HPC, where you have just some pre-processing commands that show, it tells Slurm different things about the job. Like, for example, if you want like 50 gigabytes of memory for this job, if you want to have like 24 threads, basically that's all, like all this information is in the beginning, separated by like a hash and then an sbatch command, and then you have your sort of parameter describing in like the values of those parameters. Most HPC jobs, they'll have some sort of including some sort of modules. So in this one, I'm just including star, including SAM tools, and then you'll have a reference to, like, this is an index reference of the actual genome that your reads are going to be mapping to. And here's a trick. Like if you try to run star on the HPC, multi-threaded star, you probably run into a problem where it's just giving an error. I know that, like, I've tried this before, just running on a single thread, and like single thread works okay, but when I try to increase the threads, I kept getting errors. And for me, it was the setting this parameter before running star. You limit. It's just star uses so many temporary files when you're doing this alignment, maybe 100, maybe even more, but there's a certain, like, Slurm doesn't allow a certain number of files to be open, more than a certain number of files to be open at the same time. If it tries to open like 100 files at once, it'll give you an error. It might even kick you off of your back, or it might just cancel the job. But if you set ulimit to like what, 10,000, this will probably fix that error for you. So if you're having trouble running star multi-threaded, this will probably solve your problem. Specifically, the things to keep in mind is that when we're running it in quantum mode gene counts, there's some information about the chromosome positions here too, but basically just running star, aligning the data, and running sorting it, running SAM tools to index it, and just renaming the files. So this is just an example of how you would process, and how would you work with like raw data to get it to a point where you actually are able to analyze it. So I guess that's maybe the end of the first section, where we're doing like the HPC commands to actually process data. Does anyone have questions before we move on to the section where we're actually going to be working with R to sort of wrap this all together and actually analyze it? So if not, I'll just go over some of the code. So at the end result of this, if you open up that zip file, there's a folder called data. And in that, there's a subfolder called counts_RNA. So this is an example of what you would get from the feature count, from the feature counting of running your star aligner. It's just it's a text file that just has the name of the gene and 3 columns of data. These are the number of fragments that are mapping to that specific gene location. And there's 3 columns because there's one for the total number of fragments that are mapping there, and then there are it's information on the strandedness of those fragments. Like maybe here, like 24 of those fragments map to the forward strand, 1 fragment maps to the reverse strand, and there's 25 fragments altogether that map to that. But this is the sort of process data that we're actually going to be analyzing. So now we'll get to actually, like, in order, like, if you wanted to load this up, it's generally, if you're familiar with R and if you're sort of worked with R before, Basically, you're just loading in text files. You can load those text files, those gene counts in, and sort of process them. Here's some code to do that. If you're not familiar with R, like you can you can even work along and interactively run this code. But basically, you just have this function here. It just finds all the files, the gene count files. Here it's just looping over all those files, reading them in, because it's just a it's a tab separated data. You're just extracting, basically removing all the rows that just don't have a gene name associated with them. And here it's just extracting the first and second columns, the gene name itself, and the total number of reads that are that have fallen into that sort of gene's chromosome position. So here's some of the code. Just maybe sometimes if you have 50 files, maybe not all the files have been loaded properly. This is just some code so your R code doesn't crash when it has a file that is like maybe corrupted or missing data. But the actual like binding those or sort of combining those gene counts together, basically just concatenating the columns. As long as the gene names are sort of in the same order, which they are, if you're using star to do these feature counting, you can just bind them together. And at the end of sort of running this code, you should wind up with a matrix that looks sort of like this. And this is like sort of your clean data. This is the data that we're actually going to be using to run the ATACseq2 analysis. You have your rows is the genes, and your columns are each of those samples that you're working with. And the numbers are the number of fragments that map to each gene. All right, so now we're up to section 3. So that's all how you deal with data. You have your own data set, you have your own data. This is how you process it from scratch. But oftentimes you don't want to deal with data from scratch. You want to deal with data that's been already worked with, maybe by someone else. Maybe it's already in a repository. Maybe it's maybe someone has already did this analysis and they just give you a count matrix and they want you to analyze it. So this part of the tutorial, we're just going over how to do this, like how to deal with data that's already been processed, that has already been sort of like cleaned up for you, and how to like get it ready to sort of attach or to associate the information of the experiment or whatever analysis that you're running with the actual data. So there's a repository called GEO, and this is like, it's a very popular one, and it's very easy to upload, very easy to download. There's even these like programmatic interfaces into how to actually like extract data from GEO. It was actually originated for microarray data, and for microarray data, there's actually built into the website is all these like functionality for actually to analyze microarray data. But it also has like ATACseq, ChIP-seq, like RNA-seq, like pretty much all omics data you can upload and download from GEO. And there's an R package called GEO Query, where you can use this to just programmatically just access all this information. So this section, if you can, like this is the part where you can interactively do. You can like run this. You should be able to like copy paste this code, or if you want, there's there's a file in that zip file. There's something called tutorial code, and this is a text file. It's R code that it has all the information, all the code that is in this vignette. It's already copy pasted into a file. So if you wanted to, you could just run this code line by line, or run it all together. Like you could, whatever. But all the code here, so you don't, if you don't want to, you don't have to copy paste. You can just start with this tutorial.r code. And it's sort of broken down by section, like section by section. Here is like RNA section 2.6. It's all the code from the vignette. You can run it here, like right here, we're up to RNA section 3.1. So if you wanted to, you could start from here and just run it in RStudio or however you run your usual R code. So here, this is the GEO query way to extract data. Generally, you have a project that has a session number. Usually when you're reading papers, there's going to be like a number describing where that data was uploaded, like which repository. And if it was uploaded to GEO, it'll have like a number that has sort of this format that looks like GSE and then a number. And the way you can extract that data from R is just use this function called getGEO and then the name of the number. If you want to cache that, so if you don't want to keep like this code is basically downloading the data into your laptop. But if you don't want to download that every time, you can just cache it. You have a temporary file. You can just store it to some sort of temporary folder. And if you run this command again, it'll look at that temporary folder rather than just always downloading it. So in this GEO query sort of way of extracting or downloading this data, there's a function called pdata where you can get phenotypic information about your data set. And basically it just extract like a matrix of every sample and information about that sample. Maybe like there's gender information, there's like basically all the information about that sample would be in this phenotypic data. You could be extracted from this pdata function. Here, every row here is a different sample, and every column is just information about that sample. For example, there's age, there's a tissue, like the type of tissue is, there's gender here, the type of data, like where it was prepared, the library that was prepared, like just information about that sample. Here, I'm just displaying the first 5 here. There's also, I mean, this is code, you can use pdata, or you can, like, if you wanted to actually get into the inner workings of that data frame or that data class of how it comes, like you can access it like through the actual members of that sort of that variable. So generally you can also, that's how you access the phenotypic data, but you can also access the RNA, the expression data from GEO. It's very easy with microarray data, but with RNA data and others, generally they don't have programmatic access. You actually like have to download that RNA data like separately. And here I have a link to the GEO site. So it's just an example of this is just like an example, just for this example that we're running. Generally, it has the name of the site or the paper that was associated with this data. It has some information like the submitters, the authors, various information on the samples. And then generally down here, there's some way to download the expression data, the actual data of the experiment. And here it's in a CSV file, and you can just download it straight from here. I've already downloaded it in the zip file, so you don't have to do it yourself. But if you wanted to, generally to access the data, it's from the GEO website, go to the accession number of site, scroll all the way down, and usually the information is here. So here I'm just loading it in. And here's a tip: if you're familiar with R, if you deal with R, I'm sure you all know this function, read CSV. If you use this function, it's generally like it's a slow function, and it can't deal with zip file. So if there's anything that you take from this tutorial, I suggest you use this function called fread from the library data table, data.table. And this is just like 10 times faster. Generally, I use this. I stopped using read CSV just for reading data, but it's like 10 times faster than CSV, and it can work with zipped files directly. And then if you don't really like using the data frame interface, the data table interface, you can just turn it back into a data frame. But this is like, it should run in like 5 seconds rather than like a minute or so, just to read this data. But basically, what you do is like you have your matrix, your CSV file, you load it into R, and then you have to format that so the rows and the columns of your data matrix match the rows and columns of your information matrix, your phenotypic matrix. To do that, generally, especially for when we get to the ATACseq part, we just need to make sure that you have a matrix of the counts, of the gene counts, and you have a matrix describing the information about each of the samples. And that's what this code is. And I guess I won't go over all the details, but this is something like the details are here. So like as you're sort of working with this in your own time, you can line by line work through these details and sort of copy paste, or just use the tutorial and sort of like follow along with the code to sort of process this data, so to make the columns match up. So I sort of feel like I probably spending a lot of time just on the processing. Maybe we should get to the ATACseq part of the vignette. But basically, there's like this section 3.3. It's just you need to, like, one of the things, whenever you, like, wherever you get your data from, whether it's like data generated here at Einstein, whether it's like downloaded somewhere, given to you, you always have to make sure that the columns of your matrix match up to the columns or the rows of your information about the samples. If they're like by default, generally, they're not going to match up. Maybe they have different names. Maybe you have to sort of find out like some sort of ID from the names to make sure those IDs match up. But generally, you just have to make sure that those columns do sort of align. Otherwise, all your results will be, they just won't make sense. So this code here is section 3.4. We're just, that data set in this example, it's a huge data set. It does almost like 1,000 mouse samples. And of course, like you can work with a data set this big, but it just takes so long. And for this example, we're just going to be doing like a little subset of this data. We're just going to get, let's see, dealing with mice that are just from like 2 specific tissue types and 1 specific age type. So we're just extracting a small portion of this data just to work with, just so the examples don't take like an hour to run. So here, this code is just getting a smaller subset and just working with this. And this code is just another sanity check just to make sure that the column names of your expression data match up to the phenotypic data. So here, section 4, this is the sequencing part. This is the analysis part using ATACseq. So so if anyone, I wonder, like, who here has heard of ATACseq, like, for doing this thing? It's probably like the most popular package for doing any sort of differential expression analysis. And if you've heard of ATACseq, most likely, I'm guessing maybe 90% of them. It's basically, it's the best resource there is for learning how to do this analysis. And if you are going to be doing this, it's something that you should go through. Like, you should read it over from front to back at least once. I mean, it's a long read, but there's just so much information about this. By doing this sort of analysis. What we have here in section 4 is just a very short breakdown of a quick overview of how to sort of, basically almost like a quick start, like of how to like, if you don't have the time, you don't want to read through like 2 hours of like information. This is just like a very like compact version of what's going on in ATACseq and how to use it. So basically it's you sort of like think of this analysis as like a regression. So you have your gene counts on one side, and then you have some sort of phenotypic information or some sort of data that you want to sort of, remember, experimental data that you want to see if it's related to the gene counts. And it's an analysis that's run on every single gene, like over all genes, like 20,000, 30,000 genes. But I like to think about it just first according to one gene. So you think like one gene, and you think of one experimental information, like maybe gender or so. So you have like, how does gender affect the expression of that one gene? And if you just think about it of like a scatter plot, where along the y-axis is the gene expression, and along the x-axis is like the gender, like the male versus female. Maybe the males are higher than the females for this specific gene. And that's the sort of information that you're trying to capture when you're doing this sort of differential expression analysis with ATACseq. So usually when you have data like this, if it's just like count data, usually like a t-test would do. If you have like large samples, or if you know the data is normally distributed, usually a t-test can be used. In this case, we're not using normalized or normal data or Gaussian distributed data, because we're dealing with counts. Actually, like the numbers of fragments that are mapping to each genomic position. So in that case, a t-test generally isn't a good choice to do this sort of analysis. A Poisson distribution, a test involving a Poisson test, that's another choice, because that sort of works on taps. It sort of develops for sort of count or integer counts, integer values. The thing about a Poisson is that the variance of Poisson, it's sort of just built into that distribution. The variance is equal to the mean. Like as the mean goes higher, you know that the variance of the distribution also gets higher. The mean is equal to the variance. So that's why we use this thing called a negative binomial distribution, or a test involving a negative binomial, or negative binomial regression, where it's like a Poisson, but it doesn't have that constraint where the variance is equal to the mean. There's a parameter in this negative binomial distribution where the parameter is called dispersion, where it's sort of like the variance is related to the mean, but it doesn't have to be equal to the mean. It can be over dispersed, it can be greater than the mean, or it can be lower than the mean. But there's basically, it's like a design, but with an extra parameter that allows the variance to sort of not be equal to the mean. And by using this extra parameter, it's sort of, you get a better, a more accurate picture of this sort of regression. And what DESeq2 does in this package is that it sort of calculates, or it sort of finds a good value for this dispersion parameter using information of all the genes. So it uses information on like the 30,000 genes, usually by looking at like the mean expression of the genes, and then it tries to find valid or accurate dispersion estimates for each gene that you're working with. So it's a lot more, you're using more information than just looking at a single gene. You're using information from the entire data set, like 30,000, 40,000 genes, to get like a more accurate picture, or a more accurate sort of representation of the amount of variance that's being described in this regression. So that's what DESeq2 does. It just uses a negative binomial distribution, a test involving the negative binomial distribution, to run this sort of regression test. So to actually run DESeq2, there are basically three parameters. They're called call.dat, count data, and design. So call.dat is just a matrix, and what this is just the it's a matrix that has the phenotypic information, the data describing your experiment. So the number of rows is the number of samples, and the number of columns is the number of basically experimental information about the data. This is where you keep your gender, your tissue type, your age, all that stuff. It's in this data frame called call data. And then you have count data, and this is the expression matrix. This is the matrix that describes all the genes and how many fragments are mapped to those genes. And this is a huge matrix. And just one thing to note is that the number of genes goes in the rows, and the number of samples goes in the columns. So it's actually transposed from that phenotype information. It's just something to keep in mind. It's generally a very large matrix. And then there's the design, which sort of maps the relationship between variables in that phenotypic data set to the gene expression data set. And if you're familiar with R, it uses this formula, and a formula generally has two parts, a left-hand side and a right-hand side, and it's sort of separated by a tilde. You could sort of think about this like as a scatter plot. The thing on the left-hand side is on the y-axis, and the thing on the right-hand side is like the x-axis. And the thing about a formula is that it doesn't have to be restricted to a single variable on the right-hand axis. This is where you describe your regression, your regression model. You can have more than one. You can have multiple covariates or multiple variables on your right-hand side. So there's something about the notation about that right-hand side is that you can include multiple variables with a plus sign. So if you have maybe if you want to model age and gender, you would use a plus. It also has this notation like we use an asterisk, where it sort of combines both the main effects and also the interaction between these variables. And this is just a simple, it's just notation for this, where if you wanted to include just interaction, you would use a colon. It's just that this asterisk, it's just shorthand. It always includes the main effects along with the interaction effects. But and generally to sort of use this with the DESeq2, you just insert the right hand. So basically you just specify the tilde. So you ignore the right hand side, you ignore the left hand side and only specify the right hand side of that formula. So basically you need three things to run DESeq2: your phenotype matrix, your expression matrix, and your formula. So actually running the code here is it's actually very simple. It's like three lines of code to actually run DESeq2. Well, really the hard part is preparing that expression matrix, running it, getting the data processed or getting it formatted, getting it sort of aligned so it sort of makes sense. But once you get to that part, running DESeq2, it's easy. It's basically you just run DESeq data set from matrix, this function here, and you just give it those three matrices. Here's RNA data small, the phenotype small, and the design. So here in this example, we're just running a differential expression between two tissue types, bone versus brain. So this sets up the matrix. This DESeq2 DDS that just actually runs the analysis. It calculates the dispersion parameters, runs the differential, and then results sort of just puts it into like a easy to use matrix format. And yeah, that's it for running DESeq2. It's really the hard part is actually getting to the part where you actually have your formatted clean data sets. The end result of this is it's a data frame that has every row of the data frame is the genes locations, and it has 6 columns. The first column is the base mean of the average gene expression for each specific gene. And then there's the log2 full change of the between the two groups, or if you're doing like a factor-based analysis that's between the two groups, there's log full change, it's a measure of the standard error of that log2 full change. There's a statistic, I think it's a Wald statistic of it's just a statistic representing the differential between those two groups, a p-value and an adjusted p-value. So basically, like for me, the hard part is setting up the DESeq2. I guess the easy, but like once you've set it up, running DESeq2 isn't really a big deal. And then once you have that, there's basically you can do any of your downstream analysis. Just as an example, I mean, you can do anything you want, but as an example, you might be wanting to select the upregulated and the downregulated genes, like maybe the ones, maybe we have like a threshold on the FDR adjusted p-values, and you want to select all the ones that have like higher in one group or lower in one group. There's just some code to do this. It's a way to select all the ones that are, here's just like a quick way to get the top 20 and the bottom 20. You can do like a PCA analysis or a PCA plot. This is generally useful to do before running the actual analysis, because you can tell if there's sort of maybe batch effects, or maybe if there's like a global difference in your samples, just even before you actually run the analysis or the differential. But you can do like a PCA plot where you show sort of like a sort of dimension reduced expression or a visualization, or it's sort of a way to sort of summarize the global expression across different experimental conditions. So also you can also run a something that's always done is you run a volcano plot, once you have the differential expression results, where you show it's generally it's a simple plot that shows on the x-axis like the log full change, on the y-axis some sort of a transformed value or transformation of the p-value. So more significant results are shown higher along this y-axis, and results that have a higher full change are also showed at the extreme ends of this x-axis. So yeah, so basically, I think you'll probably get the most use out of this vignette having code in front of you, being able to run it on your own, like line by line, just to sort of be able to just figure out. So basically, you can do this shows an analysis from start to end, how you can download data from GEO, run it, analyze it in DESeq2, and just some basic downstream analysis that you can do. But it's something that basically you're probably going to have to run it on your own interactively, just to get the most out of it, to get the most benefit from this. So I think there's so that's the end of the first tutorial for RNA-seq analysis. Maybe we'll take some questions before we go on to the ATACseq analysis. Yep. So when you're running with DESeq2, it specifically uses this negative binomial model to do all the distribution, to do all the regression. And one of the requirements for that negative binomial model is integer counts. So DESeq2 requires that your input matrix is just the raw data. So you don't want to feed it normalized data. It'll just, I mean, there are other packages like Lemma. There's actually Lemma that's limit counts, or I forgot the name of it, but there's other packages that sort of deal with other normalized data. ATACseq 2 is something you don't need to do that. You just give it the raw. Yep. So yeah, there's ways in autoplot. Let me just yeah, well, if you do inside R, if you do question mark autoplot, it'll bring up the help page of that specific function, and there should be parameters. You should be able to specify which parameters. So generally, if you want to, you don't. This is like, I think it's a function inside this library called ggfortify, and it's like a simplified version of PCA. It just makes it very easy to do these PCA plots. You can always run PCA yourself. There's a function in R called prcomp, and you give it the normalized count matrix. You can run PCA, and then you can plot any version, you can do the first versus the second, first versus the third. It basically, like, if you have the PCA vectors, you can plot any one you want. I prefer ggplot too, but you don't even need to use ggplot too. But this is just an example of how you do this built-in function to make PCA plots very easy to use. It also, you can color, or you can sort of change the shape or change the fill type of these things. So you can sort of it just makes it easy to do these plots. Or
I guess just to add to the question, if you're really wondering like what parameter to use, a lot of times like R has like it's built in like manuals. Oh, so like R has sort of like built in manuals, like you can put a question mark and like look around it, you know, the description for parameter is, I think it's like in R as the question mark, like the name of the function.
actually, this specific, like autoplot, it doesn't have a good documentation. But yeah, it's not. I just don't have code in front of me of how to do this. But it's generally like, usually like when I bet I can Google how to do this using autoplot to use this. I bet it's yeah. I can try that. But this is usually what I like whenever I have a question like this. I type in autoplot R. Usually it's like, let's open up a few of these. Yeah, maybe autoplot doesn't have it would take some time to figure out, but any other questions before we move on to the next section? All right, so let's get to the second section.
Ready for 5 minutes?
Yeah, that's fine. Sure.
122, you think you have enough time to get through it? We could take a 5 minute break.
Yep.
Okay. Yeah, so we'll take a 5 minute break. So people want to go through it.
We have like plenty of food, restaurants around the corner. Yeah. So it's like 1.3, that's the background like 1.8. Thank you guys.
Oh, you can say, is that the same one?
Yeah, it's part of the same, I guess. It's like,
Oh,
so there's, I mean, they use G45. You use like PR, and you can make the desired PC for XMR, and then you can the PR. So this is like PC2.
Get your phone. Anything I can let you in the meantime?
I'm good. Thanks.
Can I answer this? Wow.
Yes. All right, guys. It's 1:29. I think we're gonna get started for the session, which is about ATACseq.
All right. Yeah, we can continue right to the second lesson or the second tutorial. Inside that zip file, there's a file called tutorial2 ATACseq.html. This is the second vignette, and you can follow along, and it has the same format. The first section is going to be going over the HPC commands, how to do the processing of these raw data files. The second part is how to load those process files into R and do the differential expression analysis. So the main thing is for ATACseq, the processing is very similar. You always want to do the quality control and the adaptive trimming. The difference is that in the alignment, consider an aligner that allows for splicing between exons, because ATACseq is just looking at DNA. You don't need a specialized aligner to do this this part. So Bowtie2 is the one that I'm more familiar with, but there's BW, there's so many other aligners, and basically whatever one you're comfortable using, you can use it. It's just you don't really have to consider these allowing for splicing. So for Bowtie2, it just has here's just a section on like the little quirks for Bowtie2 is that it doesn't properly read in zipped files, and most times FASTQ files that you get are already zipped to be compressed into save space. So I like to use like process substitution, and it's just a bash sort of command or a bash workflow where you sort of like you can like on the fly convert something or like unzip it on the fly without having to create a temporary file. And if you follow this link, you can learn a little bit more about process substitution. And there's also basically like this part. This part is just using process substitution to, on the fly, uncompress your main FASTQ files. And then here, I'm just rerouting or redirecting the error into a certain text file, and then piping the output into another command that basically turns it into a binary. So the output for Bowtie2 is a text version of the aligned files, a SAM file, a SAM format. And in order to get it into BAM format, which is the binary format of that aligned file, you just pipe it into this command, samtils_view, to sort of pipe it in and just convert it to BAM pretty much on the fly. So it's basically one command that sort of pipes and redirects and sort of uses process substitution to like get a whole bunch of things done in just one command. So generally when you're doing ATAC or when you're dealing with ATACseq or DNA-seq in general, you're going to want to be sort of somehow dealing with PCR duplicates. And the software that I use is called Mark Duplicates, part of the card tools software suite. Basically, you just make sure that your data is sorted, indexed, and then you can just run Mark Duplicates directly from that file. And then once you have your aligned and your Mark Duplicate data, your BAM files, your aligned files, you can run your peak detection part of the analysis. And I use this software called Max2. It's actually originally designed for chip-seq data, and usually it requires two input files, like an input, like a something like your signal file and your target file and your like sort of background signal file, and it uses those two files to sort of determine like which are the peaks that are it's funny, but with ATACseq data, generally you only have a single file, just representing the signal, or how open the chromatin is. And there is a feature in Max2 that allows for peak detection using a single file. And in this, it uses like a window-based approach to sort of compare like the intensity of like a specific region to the background intensity along some window to determine whether or not there's a peak there. So this peak detection software, it gives you the positions or the chromosomal positions of every peak that was found. And usually what we do is you run these individually for all the samples that you have. And then at the very end, you create like a composite peak file that has all the peaks that were detected across all your entire sample. And then but before we get to there, I'm just going to show you like running the actual pipeline to do this thing on the HPC. It's the same idea as before, where you don't want to write out, if you especially have a large sample, you don't want to write out 10 times, 30 different times. So generally you want to have some sort of script that deals with your data so you can sort of cut down on the errors. And it's the same format as before. You just have a loop of all your input FASTQ files. You're extracting the paired end, or you're getting the paired sample. You're getting the sample the sample name, and you're just submitting your job. In this case, this is just another example. You don't have to save it to a text file and then run this text file separately. You can just in your loop, you can submit your jobs directly to the Slurm manager directly. I like to do it in a separate file, just so I have so I have like a document of all the things that I ran. Plus, it's just an extra thing. I like to be able to like look to see what I'm actually computing or what I'm actually running, because it's just so easy to make mistakes here. But you don't have to. I mean, especially if you're familiar with this or you have some experience, you can just in your loop just submit your data. Here's the actual script that does all this procedure. It does the alignment. It does the sorting of your BAM file. It marks the duplicates. It indexes the BAM file, and then it runs the Max2 peak finding software or Max2 peak detection. So the section, like once you have processed all your samples, then you want to get those results, the actual peaks that have the chromosomal positions of the peaks, like the chromosome, the start and stop of where the peaks were found. You want to have a composite, so you want to sort of wrap up all the peaks that were found across all samples. So you have a single file that sort of has the peaks detected over all the samples that you're working with. This is the R code that can do that. I use this library called Genomic Ranges that has a lot of built-in functionality. So you can do all this stuff pretty much automatically. You don't really have to care about like how to like sort of get intervals and get sort of intersections and stuff. They have like built-in functions that can do all this for you. So that's what this code here is doing. There's just a simple function typically just transforms your GRanges objects into a bed file, because downstream analysis typically uses bed. Well, a lot of them uses bed files to do sort of like downstream analysis. And one of the software packages that I use to do that downstream analysis is called it's part of the bed tools toolkit or BedOps toolkit. It's called BedMap. And what it does, it takes a BAM file and it takes a bed file, and it basically just counts the number of reads from that BAM file that map into every region in the bed file. It's basically the same idea as ATACseq or what we did before with the star. Basically, it's just feature counting, just using a different tool called from the BedOps toolkit. And this is basically what's going on. It's a loop over all the BAM files, extracting various information from that BAM file, just like the name of the sample. And then the actual command is called bedmap echo count fraction map. Basically just what this is, BedOps, all the tools inside BedOps, they all work on bed files. So it has a problem dealing with BAM files. So basically on the fly, just using process substitution, you can use the built-in tools in BedOps to convert a BAM file to a bed file. And then you can just do your interaction, or you can sort of do your processing from the bed files. And that's what this is going on. That's what this, it's basically saying, bed map, I want you to count. I want you to use the regions defined in this bed. And then this thing is just, it's a map. It's basically an on-the-fly map of the BAM file, converting it into a bit file, and then saving all that stuff into a file. And if you look inside this zip file, under the data folder, there's another folder called counts.atac. And this is the result of running that bed ops, the bed map. It's just, again, it's a text file. Where's it? It's a text file that has, this is the format that BedOps works, works through. It's just the chromosome, the start, the end, and then it has some columns that you can basically ignore. But the second one, it's a single element separated by a pipe. This is like the strand, which direction the reads were mapped in, and then a pipe just showing the number of reads. It's just a delimiter. But basically, what we want to do in order to use this information, we want to extract just the number of counts from this text file. And that's what's going on in this section, section 2.7. Basically, we're just loading all these files. And this is like an example of what they looks like. I just opened it up in a text editor, but here you can use R to load it up. It's just your text file. You're just looping over all the text files. Extract the last section to actually extract the counts, and then put it into a data frame. Here, just combining all those data frames together and basically creating your matrix. This is just this stuff here, just prettying up the name, because a lot of times when you're working with data or working with samples, they might have a big, long, complex name with all these like lanes and maybe like date or other information that you're not really interested in. This is just cleaning all that information. So you're just dealing with a very simple, easy to use sample name. Here, like this stuff here, I'm basically just getting an identifier for each peak region. Before each peak region is sort of identified by the chromosome, the start and the stop. And here I'm just concatenating all those together to make a single string that has just a unique identifier for each of those peak ranges. Here we're just getting rid of NA counts. And at the end of all this code, you wind up with a matrix. It's clean, a clean matrix that shows the number of peaks along the rows and the number and the samples all around here. The samples are in the columns. And now we get to actually running the ATACseq analysis. So this is your starting input, and you want to run a differential between various experimental groups. In this one, it's the control versus the drugs. So the first step is basically you need to have a matrix that describes your experimental conditions. In this case, it's very simple. The first two samples are control, the last two samples are drugs. So you can just use, for this example, it's very easy to make a data set that has this information. Basically, just this code makes that data set. It's just the first one is just the name of the sample. The second column, I call it comp one. It just shows which group each sample is made of. So the first two are in the control group. The second two are in the drug treated group. And then running ATACseq, it's the same procedure as RNA-seq. Even though it's ATACseq, it's the same procedure. You just use the ATACseq for matrix, give it the three matrices, the three input arguments, and the formula or the actual comparison is comp1. And you give those three commands, run the ATACseq, get the results, and this is your result. It's the same 6 columns. Here is just looking at the first 50 columns, but it shows each chromosomal region. It shows the base mean, the log2 fold changes, and I guess usually the most important one is this, the adjusted p-value. So that's an example of how you would run it. Generally, if you have your data, if you have a small sample size or a small experiment, you can just do this as is. But generally, you have lots of data, or you want to run multiple comparisons. So you don't want to write this. You really don't want to do this, copy paste this code and run it 50 different comparisons. Generally, you want to write a generalized function, something that wraps up everything you want to do about your analysis, and then you want to apply that function to any comparison group you're working with. And that's what's going on here. You just basically getting in all your data in this function. And basically, this is just running, it's sort of encapsulating or encompassing like a ATACseq analysis and any other information, any other thing that you want to add wrapping it up into a function, and then using that function to call, using the function call to actually do your analysis. So that's what's going on here. So basically, like this function call, it's basically doing the same thing as before. It's doing some. It's a little bit more. It's like sorting by full change. Adding some peak count information to that. But in the end, I'm like you should go over this function on your own. But running it, I think it's very straightforward. It's just the output from running this function itself. It's instead of using well, this is the output. You have like the peak D, you have the chromosome, so you have the counts of the drugs and the counts of the normal samples. And then at the very end, it's just something I added. It's something very important for doing any downstream analysis, is that you want to add the chromosomal position of the region in the format that most downstream analysis tools use. And that's just chromosome, colon, the start position, dash, and then the end position. This will make the analysis, or sort of downstream analysis, a lot easier. And we'll see that at the very last step. But just adding this chromosomal position to your routine, it just makes life easier for analysis down the line. So another thing that most people want to do is they want to annotate these peaks, because right now, if all you have is just the peak name, like chromosome 6, some number, some ending number, this isn't really very informative. So you want to sort of be able to figure out what's going on here, like which gene is this closest to. So I use this function inside of ACME. ACME is the package, and there's a function called find closest gene inside of there that can sort of append the information on the closest gene to your ATACseq results. And here I just it's just a customized function that can sort of do this stuff inside it's like a wrapper around lapply. So you can do this, like you can parallelize this. In this case, it's just doing it over 4 cores, but this is just an example of how you can parallelize a function that wasn't originally meant or built to be parallelized. You can use this function on library parallel. Basically, if you're used to using lapply, parallelizing something over multiple cores, it's as simple as just adding MC to your lapply and just giving you the number of cores. And this is just another feature that if you take away anything from this lesson, if you're not using MC lapply and you're used to using lapply, it's so easy to parallelize. It's so easy to like make use of all the resources that you have on your computer. But basically, this code will just append the information of the closest gene to your peaks that were detected, or to your peaks and the differentially expressed or differentially expressed genes. So running this append closest gene information function to your result, this is the sort of result you get. You get your gene name, you get the name of whatever some other identifier, the chromosome, the strand which is located on some information about the gene, like the transcription start, the transcriptions end, some coding region start, some coding region end. You like the ACME function append closest gene information. It has a lot of stuff that's appended to the information. And it also, since it's using the end result of that customized function, the generalized function, you also have the gene counts and also that chromosome position at the end. So here, and I think we'll probably have time to get through all this, but here, once you have these results, it's also a good idea to visualize. And one of the things that you can do is you can load in the coverage files from your BAMs, and you can use, if you downloaded IGV tools and you download it to your desktop, you can use command line tools in IGV to basically create a coverage file from the BAM file, so that you can just load into IGV. And here's some commands that you can do that. It's basically just a loop that's looping over every BAM file and just using IGV tools count. That's the actual command that you can turn it into a coverage file, which has an extension TDF. So basically, it just takes this big large BAM file, which is usually like 5 gigabytes long. It turns it into a smaller coverage file that just contains the region, or basically the fragment counts over a windowed-based summarization. And you can sort of use this to visualize your ATACseq counts. And a little bit earlier, but there's TDF files. So here's just an example I think these are just a few hundred megabytes, but they're still pretty big. But I'll give you an example of how you would sort of do this visualization. So at the end of the DSeq2 analysis, all this code here, the end result is that you have let's see, you have this this file here, and a lot of people that I work with, they're very familiar with working in just in Excel. I guess especially if you're not used to R, and to get a result file that sort of isn't that's in R format, it's very hard to work with this. But I think getting a spreadsheet like this, and this result here is just the result of running all this code in the vignette, like all the code, the previous code, append closest information, and the other generalized function. The end result is just this file here. I just call it DSeq2. I don't know, This is basically the results of your analysis. And you can open it up in Excel, and it's just a list of all the peak regions that showed significant difference between the two experimental conditions. And what you can do sometimes is you can do, there's a column called distance. So maybe you're only interested in peaks that showed a difference in open chromatin, or a differential in open chromatin that are very close to a transcription site of a gene. So then in that case, you can sort by distance and then only look at the regions that are sort of close to a gene. And we'll do that here. So we'll just sort by distance maybe. Or maybe sort or filter by like maybe 2000. So it's just a list, like a short list of genes or peak regions that show differential between the two experimental conditions. And just that generalized function, as I mentioned before, it just tacked on a little section right at the end, a column called chromosome position. And this is something that you can open up, and you can use this to navigate in any of your downstream analysis. And I'll just show an example of that. So IGV is something that you can download. You can open it up in a web browser. Just go to Google, type IGV. It'll take you to the step where you can run it in the web browser if you wanted to. But I find it's faster to actually install it on your program. And if you install it, you also have access to all those command line features. So this is what it looks like. And I'm just going to load in those TDF files just to give an example of something that how we can sort of do this. So there's 4 files, and I'm just going to call, like, these first 2 are the control samples. So I'm just going to color them. I'll just maybe color them pink, and these are blue, just to distinguish them. And the good thing about this is that you have these results. I'm just copying the chromosome position of this significant result. And maybe, yeah, so you can actually see the raw counts. You can see that the drug treated samples, the counts in this peak region, they're like, we should expect to see when we visualize this, the drug treated samples should have higher peaks in this region. So you can copy that, go right to this text input for the chromosome position, and it'll zoom you right to this area. And I like to zoom out. And what you can do is, I guess, like, as you get more familiar with these tools, it gets easier to navigate. Maybe I'm going too fast because I feel like I'm running out of time. But you can sort of check right now, like, this just shows the coverage of something. I guess it was, I guess it's not really close to it. It shows that this is one of those peaks that were detected that showed differential open chromatin in the drug treated groups. They have like a little bit higher more open chromatin. Maybe we'll show a different example. This one might be better. And if you're working with IGV, something you do, if you go right click, you auto scale, it'll sort of find the best feature for each region, for each sample, and then you want to set the data range to a specific the height. It's a sort of something that you do automatically, because by default, if you just set auto range, each sample will be sort of scaled so that the highest peak matches the height of the plot. It's just something to do. Like if you do auto scale first and then you go set data range, it automatically will sort of configure the height so it matches the data that you're working with. So this is another one of those examples where it shows higher, I guess, more open chromatin in a drug-treated sample. And just when you have a short list like this, it's very intuitive, I think, to just copy and paste this chromosomal position. Sorry, open it up and just use IGV, just open it up in these chromosomes in like the interactive view. And sort of see what's going on here. So I guess yeah. So I sort of sped through things because sort of I guess took a little bit too long, too much time in the previous part. But I guess that's the end of the lecture. But I'm willing to stay like half an hour, even an hour later. If anyone has questions, like we can answer them or ask them now. And I can go over specific things. If you wanted to go into more detail, feel free to ask.
Okay, guys. Let's give a round of applause for Dr. Like you for a lot of work putting together this vignettes. And here are some few quick announcements to make before we get to the next session. So basically, you still have like some difficulties to get the cluster ATACseq. I would strongly encourage you to reach out to Einstein IT, for example, tickets. In addition to the SPV, so this session and the subsequent sessions, the tools are actually also provided by Dr. Zheng from the genetic department. So thanks for their support. Alex, do you have any other announcements?
We're gonna post some like supplementary materials on the website. So if you are like thinking more about these things, you can check the website. It will be updated continuously. And additionally, once the recordings are available, we'll post information on there on how to access the recordings for the session.
Thank you for